Hello, we from Group 4. We will discuss about histology of fish tissue.
Introduction. Histology is a vital branch of biology that focuses on the microscope structure of tissue. In aquatic science, histological techniques are essential for examining the internal feeder of fish and other aquatic organisms. This technique allows scientists to identify normal tissue structure and detect any abnormalities, infection, or damage caused by environmental or physiological factors. By analyzing tissue samples at the cellular level, researchers can gain valuable insight into a health, function, and the development of aquatic species. This is particularly important in fisheries science, aquaculture, and environmental monitoring. As such, histology research serves as a powerful tool in supporting biological research and conservation effort. This experiment aims to introduce study to the fundamental method of histological sample preparation using fish tissue. The main purpose was to provide hand-on experiment in collecting, fixing, embedding, sectioning, staining, and mounting tissue samples to prepare them for microscopic analysis. The scope of this mini project includes the use of various fixatives and staining techniques to preserve and enhance the visibility of tissue components. These steps are crucial in producing high-quality histological slides that can be used to study the fine details of cellular and tissue organization. By completing this experiment, students were expected to build a solid foundation in basic histology. Through the project, students worked with various types of fish tissue such as gills, liver, heart, and intestine. Each step in the histological process was carefully followed to ensure proper preservation and visualization of tissue structure. The use of fixatives like formalin and alcohol formalin help prevent tissue decay and maintain cell integrity, while staining with hematoxylin and eosin (HNE) enhance the contrast between different cell types. This procedure made it possible to cleanly observe tissue organization and morphology under the microscope. Understanding the rationale behind each procedure help reinforce the importance of careful preparation in producing reliable scientific results.
Material. There are three types of material that being used. Firstly, biological sample. Fish species, Pristolepis fosciated, and other species. And second, a larvae, Claris spp, a catfish species. And the next material will be reagent and chemical. One is 10% paraffin neutral formalin. Second, internal. Third, xylene. Fourth, paraffin wax. 5. Hemostolin solution. 6. Eosin Y. 7. DPX. Next is instrument and tools. First is dissection set. Second, digital balance. Third, ruler or measuring board. Fourth, dissecting tray and board. 5. Tissue sample vials. 6. Permanent marker. 7. Paraffin embedding. 8. Hot pellet or water. 9. Microtome. 10. Glass microscope slide and cover slip. 11. Slide rock. 12. Slide warmer. 3. Coplin jars. And lastly 14. Blotting paper. Next, we will go into method. Firstly, the collection of the sample. Pristolepis fasciated specimens were used for tissue collection. The fish were immersed in ice water, a method commonly used in aquatic research to minimize suffering while preserving tissue integrity. Prior to dissection, basic morphomeric measurements including body weight, total length, and standard length were recorded for the fish. Dissection was performed using a sterilized dissection kit on a clean tray. Small tissue sample, approximately 3 to 5 mm in thickness, were collected from the heart, liver, gill, gonad, skin, and intestine. Second, dissection of the sample. Immediately after dissection, tissue sample was fixed in 10% buffering formalin to preserve circular architecture. Each tissue was placed in a properly labeled plastic vial containing a sufficient volume of fixative, maintaining a ratio of 1 part tissue to 20 part fixative to ensure complete penetration. Level included sample type, organ, and date. Dissection was carried out at room temperature, 25 degrees, for approximately 10 to 12 hours. Buffered formalin was used due to its ability to preserve protein and nucleic acid while maintaining pH stability, which minimized shrinkage or distortion of tissue during processing. Dehydration of the sample. After dissection, tissue sample underwent dehydration to remove water, which is incompatible with paraffin wax used in embedding. Dehydration was achieved by transferring the tissue through a graded series of ethanol solution, beginning with 50%, followed by 70%, 85%, 95%, and finally 100%, with each step lasting 1 hour. This gradual process prevented osmotic shock and tissue shrinkage, ensuring preservation of structural detail. The dehydration was conducted at room temperature in closed container to prevent ethanol evaporation and contamination. Clearing method happened once dehydration was complete. The tissue were subjected to a clearing process using xylene. Clearing serves to remove ethanol and make tissue transparent by replacing alcohol with a substance that is mixable with both ethanol and paraffin wax. The tissue were immersed in xylene for 2 changes, each lasting 30 to 60 minutes. Xylene not only increased transparency of tissue but also enhanced paraffin infiltration in the next step. Equipment ventilation and personal protective equipment were used during this step due to xylene toxic and volatile nature. Impregnation with paraffin. Cleared tissue were infiltration with molten paraffin wax maintained at a temperature of 58 till 60 degrees. This step was crucial for replacing the clearing agent, which is xylene, with paraffin, a medium that provides structural support during sectioning. The infiltration process involved 3 sequential immersions in fresh molten paraffin, with each immersion lasting approximately 1 hour. This multiply change ensured the complete removal of residual xylene and allowed thorough saturation of the tissue with wax. Care was taken to maintain the wax temperature slightly above its melting point to avoid overheating, which could have led to tissue hardening or structural damage. This impregnation step made the tissue firm enough to withstand microtop sectioning without distortion or collapse. Embedding. Once paraffin infiltration was complete, the tissue was immediately embedded in metal mold filled with fresh molten paraffin wax. Proper embedding was crucial to preserve tissue orientation and ensure that desired anatomical structure will be visible during microscopic examination. Each tissue sample was carefully positioned in the mold to ensure correct alignment, such as placing epithelial surface or cross-sectional plane in optimal cutting position. After orientation, the mold was placed in cool water to rapidly cool and solidify the wax. This rapid cooling helped prevent paraffin crystallization and ensure the formation of firm, homogeneous block. The hardened paraffin block provides a stable medium for subsequent sectioning using the microtop. Cutting. After embedding, the paraffin block containing the tissue was trimmed to expose the surface of specimen. The block was then secured in the holder of a rotary microtop, ensuring it was tightly fixed and properly aligned for uniform sectioning. A clean microtop plate was installed and adjusted, and the clearance angle was set to approximately 3 to 4 degrees to achieve optimal ribbon formation and minimize tearing. Initially, the section thickness was set to 50 micrometers to trim away excess wax and fully expose the tissue surface. Once the tissue became visible, the section thickness was adjusted to 4 till 6 micrometers for standard histological sectioning. Thin ribbon of section were produced by rotating the microtop handle in a consistent and controlled motion. To access with ribbon formation, especially when the wax was too brittle or soft, one edge of the block was sometimes gently rubbed with a finger. The resulting ribbon were carefully transferred into a warm water bed, maintaining at approximately 45 degrees to flatten them and remove wrinkle. Sections were flooded with the shiny sign up, and float were removed using fine forceps or a seeker. Once flattened, individual sections were picked up using a sieve-coated glass slide. The slides were gently lifted from the water, and excess water were removed using blotting paper. Finally, the slides were dried in an incubator set at 60 degrees to 20 to 60 minutes to firmly enhance the tissue and ensure the slides were ready to standing. Staining was performed using the Hemostaxylin and iosine technique, which allow clear differentiation of tissue structure. First, the sections were deparamerized and rehydrated by passing them through xylene, 3 washes of 1 minute each, followed by descending concentration of internal 100%, 95%, 85%, and 70%, and finally rinsed with tap water. Hemotoxylin was applied for 2 minutes to stain nuclear blue to purple. Excess stain was removed by rinsing with tap water. The slides were then treated with 70% and 95% ethanol before counterstaining with iosine Y for 1 minute, which stained cytoplasm and connective tissue pink. Final dehydration was achieved using grinded alcohol 95% and 100%, followed by xylene to clear the stained tissue. Mounting. Finally, the standard section were permanently mounting using DPX mounting medium. A few drops of the resin were placed on the standard tissue section, and the cover slip were gently lowered to avoid the formation of air bubble. The slides were placed on a slide warmer and let to dry for at least 24 hours, allowing the mounting medium to harden fully. Once dry, the slides were ready for long-term storage and microscopic examination. This final step preserved the standard tissue and ensured its optimal clarity under the microscope. Microscopic examination. The prepared slides were examined using a compound light microscope. The slides were initially observed under low magnification to locate the tissue section. Once key areas were identified, higher magnification objectives were used to examine circular details such as nuclei, epithelial lining, and tissue boundaries. Focus adjustment and fine-tuning of the condense and light intensity were performed to enhance image clarity. Each tissue was scanned systematically from edge to center to ensure complete evaluation. Observations were recorded by identifying prominent structure, comparing staining pattern, and noting any artifact or abnormalities. Next is we go into the result. The first image shows heart tissue from a zoom out using low magnification view. The zoomed out view of the heart revealed the general organization of its cardiac tissue. The compact and spongy myocardium was distinguishably forming a dense marshal wall around the heart chamber. The pericardial cavity surrounding the heart was also visible. This structure aligned with typical Taylor's heart anatomy, where muscle lies provide strong contraction for circulation. Next, heart tissue in zoom in with high magnification. At higher magnification, strengthened cardiac muscle fiber with central located nuclei were visible. Intercalated with enable synchronized contraction were distinguishable. The well-organized muscle bundle are typical of healthy myocardium in teleos. Next, tissue is gonad, which taken from the high magnification. The gonad section display cluster of developing germ cell. In testicular tissue, various stages of spermatogenesis were likely present, including spermatogonia and spermatozoa. If ovarian, different oocyte stages such as primary and vitellocytes may have been observed. This reflects the active reproductive phase, consistent with Taylor's protective histology. Next result is gill from a zoom out using low magnification. Primary gill filaments were evident, bearing secondary lamellae responsible for gas exchange. The filament architecture was aligned in parallel row extending from the gill arc, resembling standard gill anatomy in fish. Gill in zoom in using high magnification. Secondary lamella were lined with squamous epithelial cell, enclosing pillar cell and capillaries. Chlorine and palmon cell involved in osmogulation may have been seen in the gill epithelium. This is consistent with the high specialized morphology of fish gill for respiration and ionic exchange. Liver in zoom in using high magnification, the zoom in view revealed phylogonal hypocysts with central nuclei and eosinophilic cytoplasm. Sinusoidal capillaries were visible between hypocyanid row. This feature aligned with healthy hepatic tissue in fish, allowing efficient metabolic exchange. Liver in zoom in. Using high magnification, the zoom-in view revealed phylogonal hypocysts with central nuclei and eosinophilic cytoplasm. Sinusoidal capillaries were visible between hypocyanid row. This feature aligned with healthy hepatic tissue in fish, allowing efficient metabolic exchange. The next tissue is skin. From a zoom out, the skin showed a distant epidermal and dermal layer. The dermis likely contains scale pocket, permanent cell, and collagen fiber. This layered structure is common in scale thalamus species. Skin in zoom in. Stratified epithelium was present in epidermis, along with mucous cell interspersed between cryotyrosine. The dermis featured fibrous connective tissue supporting blood vessel and chromatopores. This finding reflects typical fish skin histology, a depth of protection and pigmentation. Next is intestine, has been taken from other group, which is taken in low magnification. This intestinal section reveals prominent musculous fold line with columnar epithelial cell containing goblet cell. This structure aids in nutrient observation and mucus secretion. The intestinal architecture matched that description in Thalia's digestive anatomy. Lastly is catfish larvae, also from another group. In low magnification, the larvae tissue section show early development of vital structure. Eye with visible lens, neural tubes, notochord, and stomias were present. The developing gut and musculature were also observable, reflecting normal organogenesis stage in Thalia's larvae.
Discussion. Cracking in the tissue. Some of the tissue are cracking and the potential reason of the cracking are over-dehydration with alcohol after fixation, cutting too fast, block is too cold, and potential bubble in paraffins. Ways to overcome. Carefully optimize and monitor the time spent in alcohol solution. Slow down sectioning speed. Do not let sit on ice too long and likely warm the block. Agitation of the tissue while embedding may remove any bubble from the cassette. Holes on paraffin during cutting phase. During the cutting phase, we discover holes on the paraffin wax. The potential reason of the cracking are rough trimming and hard tissue. Ways to overcome is trim it slowly and let the tissue soft a bit but not too soft. During the cutting during the cutting phase, we also lost the catfish larvae, which is why we use samples from different groups. Hard tissue. Despite the sample were cracking, we were able to observe sample. The hard tissue are in a good condition as we are able to see detail on a zoom-in image. The reason why the tissue are cracking and are from handling error. During the cutting process, we discover some holes on the paraffin wax, which is located at the hard tissue. Gills tissue. We were able to obtain the gills of the fish we used. However, due to handling process, the gill tissue samples were cracking and damaged. We were only able to observe partially of the remaining sample as only partial of it can be seen under microscope. Gonad. We successfully obtained the gonad from the fish. The gonad we obtained is an ovary which contains the eggs of the fish. We can conclude that the fish is female and mature enough to develop eggs. We were able to observe the egg tissue under the microscope. Liver tissue. We were able to obtain the liver and observe it. Despite there's a few cracks on the sample, overall, we can observe the detail on the higher magnification power. Skin tissue. Despite the cracks, we were able to observe the sample. However, we did not be able to observe the inner part of the skin and observe how deep the skin and flesh is. This is due to the placement of the fish skin during the embedding phase. Hence, we can only able to observe the outer layer of the fish skin. Intestine tissue. During the fixation phase, we did a mistake and did not remove the outer tissue that surrounded the sample. We did try to put paraffin wax on it, but it failed. Due to that, we were unable to obtain the intestine of the fish, and using another group fish with the same species to be observed. Catfish larvae. We were unable to obtain catfish larvae due to a mistake occurred during cutting phase. We were accidentally cut too much and lost the sample, which is why we observed the sample from a different group. Effectiveness of chosen fixation, embedding, staining, and mounting technique. Fixation used 10% neutral buffer formalin, stabilized tissue by cross-linking protein, preventing decomposition and maintaining cellular architecture. Embedding. Support tissue for slicing, commonly with paraffin, ensure that tissue are adequate support for thin section without distortion or loss orientation. Staining. It enhances contrast and highlights cellular components with hermatoxylin staining, nuclear blue, and eosin staining, cytoplasm pink, therefore facilitating clear visualization of tissue structure. Mounting. With suitable media and cover siping protects the stained tissue, preventing degradation and ensure long-term preservation and clarify under microscope. Advantage and limitation of different staining methods. Hermatoxylin and eosin. The usage is to generate tissue morphology. The advantages are simple, fast, widely used, clear nuclear, and cytoplasmic contrast. The limitation is lack specific for certain cell components like lipid or carbs. Periodic acid shift. Usage is for polysaccharide, mucosal substance, and basement membranes. Its advantage is to highlight glycose, mulching, fungi, and basement membranes. And the limitations are does not clearly stain nuclear or connective tissue. Manson trichome. Its usage is for muscle, collagen, and fibrin. The advantage is to differentiate connection tissue from muscle and epithelium. The limitation is complex procedure, less detailed cellular feature. Silver stain. Its usage is for reticular fiber, fungi, and basement membrane. Its advantage is highly specific and high sensitive for reticular and fungi. The limitation is technically difficult, prone to artifacts. Immunophytochemistry, or IHC. The usage is specific protein using antibodies. The advantage is highly specific and detect antigen not visible with a routine stain. The limitation is expensive, time-consuming, and require technical expertise.
For conclusion, this histological mini-project was provide valuable insight into the fundamental technique required for preparing and analyzing fish tissue at microscopic level through the systematic execution of tissue collection, fixation, embedding, sectioning, staining, and mounting. We gain practical exposure to standard procedure integral to histological analysis. This process enables the visualization of cellular and tissue architecture in aquatic organisms. reinforcing our understanding of their internal anatomy and physiological adaptation. The application of histological technique in biological research is indispensable, particularly in aquatic pathology, where perceived tissue assessment is critical for diagnosis disease, evaluating environment impact, and supporting fish health management. The ability to detect cellular abnormality of structural change through microscopic analysis contributes significantly to both ecological monitoring and the development of sustainable aquaculture practice. The findings underscore the potential of expanded research using methodological approaches to investigate a wider range of aquatic species and pathological conditions. Moreover, this technique serves as a foundation for more advanced diagnostic and research applications, include histopathology, toxicological study, and development biology. Future investigation may further refine this method or integrate them with molecular tools to enhance the resolution and interpretive power of aquatic tissue analysis.